Good evening from Singapore. To everyone tuning in, I would like to extend a warm welcome. My name is Juliana Chan, and I am the founder and publisher at Wildtype Media Group. We are the publishers of Asia's leading science magazine, asianscientist.com, and you can find us in print, online, and on social media. Our distinguished guest for today is Professor Sir David Lane, the Chief Scientist of Singapore's Agency for Science, Technology and Research. Okay, and now, to provide some background on Sir David, not only is Sir David the Chief Scientist of Singapore's Agency for Science, he is also the Chairman of the Board of Trugai Antibody Research. Knighted in 2000 for his contributions to cancer research, Sir David is one of the scientists credited with the landmark discovery of the p53 cancer gene in 1979, which he nicknamed the Guardian of the Genome. By the way, as a science communicator, I must say that is a brilliant nickname. Sir David is an expert on everything antibody related. I'm sure he dreams about antibodies sometimes, from antibody generation and characterization to antibody-based therapies for research and industrial use. And I think all of that knowledge will come in handy today for our discussion on antibody testing, antibody drugs, and immunity certificates for COVID-19. So I'm going to invite David onto the screen, and I'm going to say a big hello. Hi, David. How are you and your family doing? How is Birgit doing?
We're doing fine. Thank you very much, Juliana. I mean, we're missing our family. Everybody's in the UK and we're here. And I guess many other people are in situations like that. So it's a difficult time in some ways, but we're doing fine. And of course, there are 90,000 papers on P53 to read and a lot more to read on COVID. So I'm kind of busy as a scientist.
That's good. That's good right now. How about the P53 lab? Is it business as usual, or have you been unable to return to the lab right now?
Yeah, we aren't able to go into the lab because of the circuit breaker here in Singapore. So we have just one or two people to go in to check on liquid nitrogen in the freezers, but that's it. So all of us are busy having lab meetings. We have a lab pub quiz. We're trying to write up all our papers and
That's
also
true.
come up with good ideas for what we'll do when we get back. So it's busy, but it's different.
You know, as the chief scientist of A*STAR, Singapore's Agency for Science, you have a bird's eye view of the research taking place across all 18 research institutes. So would you be able to share with us some of the COVID-19 research taking place at A*STAR right now?
Yes, certainly. I mean, the Singapore response in general has been pretty amazing. And I guess it's one of the benefits of the investment the government made in biomedical research, around the time that the biopolis and everything started. So now we're seeing those benefits. And so in A*STAR, there's a lot going on. I mean, I guess one of the principal things has been the collaboration between A*STAR, EDDC, Tang Tok Seng Hospital to come up with the Fortitude kit. And this is a PCR-based, real-time PCR-based kit to detect COVID. It's proved to be very sensitive. It doesn't have cross-reactions with other viruses, and it's been rolled out across 13 hospitals and private labs in Singapore. And actually, Singapore government's exchanged it and given it to other countries. So more than 20 countries, including New Zealand, UK, etc., have had it, and it's been given to people in the Philippines, Indonesia. So it's been a big success. And building on that, of course, we have to keep developing the technology. So a big area is in diagnostics, and we have a DXD hub, a diagnostics hub that's able to produce and check these kinds of detection devices for their accuracy and reproducibility. So that's a big area of activity, and there's lots of innovation there. I think we all think that testing is going to be the future. I mean, this virus is not going to go away quickly. We all would want it to, but I think realistically, realizing, only 3.7 million people have been exposed, have had it, and there's 7.8 billion people in the world. So there's a long journey here. And I think testing is going to be vital. So trying to develop that, trying to get more accurate and faster testing is very important. I guess part of that is dealing with the number of genomes. So everybody's sequencing COVID. So we have Sebastian in BII who's leading the A-STAR initiative there, and he's working through this global initiative for sharing all influenza data, which is a common global site that allows all the COVID data to be put in. And it's very fascinating. You can see the variants coming and going in the different populations. So it gives you a lot of sort of molecular evolutionary history and very valuable epidemiological information. So, that's another big area. And that's important because the PCR test must be always working. So we have to keep account of any new strain variants that are coming and make sure that the bits of sequence we're using to identify the virus haven't changed. And then on another level altogether, the Science and Engineering Research Council, along with lots of other people in Singapore, are working on, producing masks, designs that you can make at home, studying how masks work. And they have a fantastic high-speed camera that can film a sneeze, and it's pretty amazing, and sort of done a lot of very good physics and maths around that to realize that the 1 meter, distance is right because the droplets evaporate, and so it doesn't go as far as some people have claimed. Very important work, and then associated with that is, I guess we're beginning to think about the long-term need to be self-reliant. So can we manufacture masks and particularly visors in Singapore? So my son in the UK has been involved in a kind of UK-wide effort where they've used 3D printers, and the same thing's happening in Singapore. So can we design and make PPE here? I guess in the long term, we're also thinking about long-term resilience. So we don't want to run out of any of the components we need for these kits. So that's all kind of being built up. And then, of course, therapy. So, I started ETC, as it was then called, the Experimental Therapeutics Center, now called the Experimental Drug Development Center. And they're busy trying to look at existing molecules, trying to come up with new molecules. And then the Immunology Network with Lisa Ng and Laurent is very, very active. And they are, great experts in the immunology of viruses. And Lisa's been working on it for a long time. And they've written several very good reviews, and they're sort of very, very busy advising and helping people to tackle this huge problem. So I think it's been exciting in a way to see everybody coming together. There is a kind of a bit of a wartime spirit, and I think actually we need that because we have to all work together more than ever to do this across the whole of the government and all the institutions to do the best we can.
Okay, thank you, David, for the update from A-STAR. So David has kindly agreed to guide us through some of these very important concepts with the help of some visuals, which I will be showing you right now. Off to you, David.
Yeah, okay. So, there's 1,000 of you or more online, and some of you are going to be much better immunologists and infectious experts than me, and others of you really will need some help. So for those of you who know everything, just be a little patient. This is just a kind of picture of a virus infection, which WHO has been using, and it's valuable in the sense that it gives us an overall picture. So the sort of shading in the background is the time that you feel sick. So in this particular virus infection, you're feeling most sick at about day 10. That's the top big, thick blue line. And that's when the virus is detectable and it's peaking. And as you can see, eventually the virus clears in this patient who's going to recover. And then you can see the innate immune response, which is right at the beginning, and that's in the thin blue line. And that then develops further. So this is the initial reaction that the body has to the infection. And this can be the release of a lot of cytokines that stimulate the immune response. And then we see the beginning of the cellular immune response, a specific immune response, as we call it. So this is where we're beginning to make T cells and other cells that are specific to the virus. And that's in the orange line. And then very importantly for our discussion today, we start to see the appearance of antibodies. So these are the molecules that circulate in the blood and can bind to and inactivate the virus. And in the serum, we see two types, principally the large antibodies called IgM, and they are associated with what we call the early response, and that's the black line. And then in the, again, think purple thick line is the IgG antibodies, And these are the ones that come in later in the immune response. They tend to be very specific and very potent. And very importantly for the discussion, as you can see, they persist in the serum long after the virus has been eliminated. So the importance of this is the understanding that serology, that is the measurement of antibodies in the blood, can give us a more persistent recognition of somebody who's been infected. So if we just measure the virus, is what the PCR test does, as somebody gets better, the virus disappears. In fact, that's how we decide in Singapore to discharge patients, because they become virus-free. But in the population as a whole, we'd really like to know how many people have been infected, because the data coming out from all over the world suggests that up to a quarter of people who are infected show no symptoms. And so this is obviously a big problem, because asymptomatic individuals can spread the disease without realizing it, completely guilt-free. But we need to realize that this is a huge issue. So at the moment, of course, we're using social distancing. We're just essentially assuming that everybody's infected as a way to get a grip on this disease. But in the long run, we would like to understand much more about the disease and much more about what we can do to manage it in society. So if I go to the next slide. This is just talking about how we measure antibodies. And obviously there are a variety of different approaches we can take. And I'm going to go through them all a little bit in detail, because they have important differences. So I'll talk about their broad properties, and then we'll show some slides to illustrate how they work. So most important in a way is the rapid diagnostic test. And this is very important because it could be done at point of care. In other words, you don't have to go off to a lab to do this. It can be very fast, and it can be done at the time. For example, persons are queuing up for an airline ticket. It could be done. It's very, very quick. Most of you will be familiar with it, because it's basically the same principle as we use for the pregnancy test. So a fast, very cheap to do, very effective kit that can be manufactured easily in large amounts. But as we'll see, there are some caveats, so we'll come back to that. The enzyme-linked immunosorbent assay, or ELISA, is a next generation assay. It's more quantitative. It's done in a lab. And the difference here is, rather than a plus minus, it can tell you how much antibody you've got, a quantitative test. And then finally, the sort of gold standard is to say, well, I've got the antibody, but is that enough? Is it doing anything? And does it matter? We'll come to this point right at the end of the discussion, because it's important. So the final test you can do with a virus is to say, can the antibody stop the virus being infectious? Can it actually inactivate the virus? And we call this virus neutralisation, because we've neutralised the infectivity of the virus. So these are the three tests. And if we go to the next slide, I'll just talk a little bit how they work. So the rapid diagnostic test, or lateral flow test as it's sometimes known, very, very clever assay. So essentially, you just take a tiny drop of blood, and it can be whole blood, which is what makes it so easy. And these molecules will then migrate up this media, this usually paper or some other substance, and they encounter different things as they migrate. So first of all, in what's called the conjugate pad, they encounter antigen, that would be in our case COVID-19 proteins, and also there's in that pool is a control antibody, which we're going to use to check that the assay is working. As the antibodies diffuse past the test line, they are captured by this purple antibody. So it's an anti-antibody. Sorry, that's a bit complicated, but if it's anti-human IgG or anti-human IgM, for example, we can capture that. And because the antigen has been made colored using gold, we get a line, so we can see a positive test line. Now, we want to make sure, in case we got something that doesn't seem to contain antibodies, that actually the test worked. So the way we do that is to have a control line, and the other labeled protein, in this case it's a gold-tagged antibody, will stop at that control line. And so you can get the readout that you can see in the bottom. So the molecules flow up the test, and then we capture the antibodies from the person who's made antibodies to COVID at the first line, and the second line just tells us that the test has worked. So if we look at a positive result, we should get two colored lines. The first line is the test line, and the control line is the check that the assay's worked. And then a negative result, we have no test line, we have the control line. And if something's gone wrong with our kit or our sample, then we can get these two bad results, which are the two that are on your right, inconclusive test. Either nothing happens, or bizarrely enough, the test happens, but the other one doesn't. So those we have to reject. So these tests are as I have explained, very commonly used, can be very successful, but they need to be very, very carefully produced and validated. And we'll come back to that when we talk a bit more about these problems. If we go to the next slide, yeah, so this is the ELISA assay. This is usually done in a 96 well plate. So this is a sort of plastic plate that contains 96 little test tubes, if you like, and we coat the bottom of the plate with antigen. So in this case, it would be one of the viral proteins, the COVID spike antigen, for example, or the N protein. And then we incubate it with a blood sample. But in this case, we can dilute the patient's blood. So we can give, for example, a 1 in 10, 1 in 100, 1 in 1,000 dilution of the patient's antibody. And then we detect that antibody binding to the COVID antigen with another antibody that's been labeled with an enzyme. And this is the one that's shown here in red. And then we add the substrate. And this is colorless. And when the enzyme acts on it, it changes color. And so we can read this very easily by a color change. And as you can see in this example, the patient at the top has a rather weak response and doesn't give much color change. And maybe their antibody would only work at 1 in 10 dilution. Whereas a patient number 2, you can see, has a lot of the red antibodies. They create a very strong enzyme reaction. And for example, this antibody might titrate out to have a titer of, say, 1 in 1,000. And that's very important. We might find in the future that to have a good protective immunity, you need to have a certain level of antibody. So this kind of test is very important in development. We may later be able to adapt the simple lateral flow assay to give us a measure of the kind of antibody response we want. So, but the ELISA assay will inform that kind of data. And then in the third example, next slide, please. This is the virus neutralisation assay. So this is what I was talking about. This is where you actually take the serum containing antibody and dilute it. And you can see here we've diluted it 10 to the minus 2, so that's 1 in 100, 10 to the minus 3, 1 in 1,000, 10 to the minus 4, 1 in 10,000. Or we've added no antibody at all, and that's obviously the control. And then to each of those tubes, we add the same amount of virus, and we incubate them together. And now we've got to do a test to see whether the virus still works. Can it still infect cells? And that's what's shown at the bottom. When viruses are plated on cells that they can kill, they can form little holes in the monolayer. We call them plaques. And that allows us to show that the virus is infectious. And so what you can see is when we have the virus alone, we get lots of plaques. And obviously this has to be very carefully run in the lab to make it work properly. And then as we dilute, you can see the antibody is working at high concentrations. There's almost no viral plaques. At 10 to the minus 3, there's a few, 10 to the minus 4, a few more. So this gives a very quantitative output of how good the antibody is at stopping the virus infecting cells in culture. The issue I would say here, obviously this is a big step up from either of the previous 2 tests, but it still doesn't necessarily mean that the person is going to be safe from infection. There was a lot of work done on Ebola virus antibodies recently, and a big comparison was done, and quite paradoxical results can happen. So in general, it was true that antibodies that neutralized the virus worked very well in preclinical, in animal models, but there were a few exceptions. So there were some antibodies that didn't neutralize in this kind of cell-based tests, but did work in protecting the animals. And there was also the reverse. So 1 or 2 antibodies that seemed to neutralize in this tissue culture assay, but weren't effective at providing protection in the animals. And this of course, is something that needs to be investigated very, very closely as we try to move towards the antibody drugs that I'll talk about later. So that's the 3 sorts of assays that can be used. And of course, people are very imaginative, and these assays can be miniaturized and made to go faster, and all sorts of clever ideas are being worked on to increase their throughput and accuracy. But at the moment, I would say the big problem is the accuracy. So the issue that's been seen, particularly with the lateral flow tests, is that they're giving different results. So basically, there was a nice article in Bloomberg. A young woman journalist went around 4 labs in London had a sample taken, and then 2 said she had antibodies to COVID-19 and 2 said she didn't. So that tells us what a long way we've got to go. We have to get these tests to be much, much more accurate. Because clearly, as we'll discuss in a minute, if you give the wrong answer, it's pretty bad. So if we could go to the next slide. Next slide, please. This is sort of trying to, and this is using WHO's advice, trying to kind of pin down what you actually know. So if you have an antibody test that's positive, then you know that the person was infected in the past. And probably if they've got both IgM and IgG antibodies, you know it's a recent infection. If they're IgM negative but IgG positive, it's probable that the infection occurred some time ago. And how long antibodies can persist after infection is a very important issue. We know it can be for at least for several weeks, and it probably can be for longer. So that's a kind of very important epidemiological range. What it doesn't tell us is if the person recovered. We know sometimes people can have persistent infection. It doesn't really tell us how potent the antibody is. We're not doing a titration here. We're not doing a dilution series. And it certainly doesn't tell us if the antibodies are neutralizing. So it's useful, but it has these restrictions. And then if we go to the next slide, the negative antibody test tells us that, well, it could say the person's not been infected, but it could also say they were very recently infected, because remember right at the first slide that these antibodies don't appear until sometime after the infection. Most strangely, I guess, it could be the person was infected but cleared the virus without mounting antibody response. So we know from some of the data in China that such cases have been reported. That is, people who appeared to be PCR positive, recovered PCR negative, but no antibody found in their blood at this time. And that could also be because the levels of detection are not sufficiently high. And it certainly doesn't tell us if the person is susceptible to infection. So this all sounds negative, but I want to come back and say that, you know, we have to be aware of all these things. But on the other hand, serology can be very, very helpful in helping us to understand epidemic and to begin to pick out people who have been exposed. So there are these caveats, they're important caveats, but they are not, they don't mean that doing this is a waste of time. In fact, if it becomes very well validated, it can become an enormously useful tool. If we go to the next slide, I want to talk now about the other kind of use of antibodies in the whole COVID-2 crisis, and that is the use of antibodies as drugs. Now, those of you who are interested in drug discovery, drug development will realize that in the last 10 years or so, antibodies have become very, very important class of drugs. So we used to mostly use small molecule drugs, but antibodies now have become very important. The best-selling drug in the world actually is Humira. It's an antibody drug that inactivates a TNF, and it's used for the treatment of inflammatory conditions. So these drugs, and many of them, of course, in cancer field, my own field, like Herceptin, are very, very powerful, very useful molecules. One's even been invented recently by a group in Sugar and Roche that actually can treat hemophilia. So they've become very flexible, very powerful molecules. And in the case of COVID-2, we only have 2 classes of antibody drugs that are potentially useful. So in the next slide, the first class in the next slide is antibodies that can help with the clinical manifestations of the disease. So I expect all of you are aware that this COVID-2 virus, this COVID-19 syndrome, can be lethal. And the lethality is really associated with either a cytokine storm or with a failure, respiratory failure. And both of these are associated with inflammatory responses. So in the clinic and approved for other indications are a number of antibodies that can inhibit or block these cytokine storms. So that the 2 first antibodies against interleukin-6 receptor pathway, these have proved to be very useful in damping down cytokine storms. I mean, they were originally developed for the treatment of inflammatory diseases, but they've been useful, for example, in children receiving CAR T therapy, where they sometimes get a big response. So they're tested and safe drugs, and their trials are happening now. They could have a big impact if they really, really could reduce the number of people who went into very serious illness. It would relieve a lot of pressure on the health system and could be very important. And then there are other approaches, which I won't go into, which are again designed to damp down and modulate the immune system. And I think these are all very promising approaches, and the trial results should come out quite soon and could have big implications. If I go to the next slide. The next slide is really a very exciting area, which I'm very keen on that, to be honest, and I should lay my cards on the table. So this is the idea that you could make an antibody that would be able to prevent infection by the virus. So this would be an antibody that would be prophylactic. You would give it to people and it would stop them getting infected, and also potentially could be used in treatment as well. So this kind of approach is known to work. There's one approved medicine, which is for the treatment of a childhood respiratory virus, where an antibody therapy is given for respiratory syncytial virus. So the model is there. And in the Ebola crisis, people started to use this approach as well and showed considerable success. So particularly Regeneron and other companies were involved in that. So all of these companies and many others now are trying to make a neutralizing antibody that could be given to patients and would allow them to speed up their recovery or protect them against infection. Now, antibodies can have very long half-life in the blood, so it's possible to imagine in the ideal circumstances that a relatively small subcutaneous injection could protect somebody for several months from COVID-19. So these could have a big impact for healthcare workers and in other areas. In fact, it's important to think about that a little bit, because when we think about a vaccine, not everybody will respond to a vaccine. So older people, people who've had, for example, their spleen removed for other reasons, or people who are on chemotherapy, or people suffering from immune deficiency syndromes, all of those people will not get protection from a vaccine. So again, this kind of antibody approach essentially substitutes for vaccination in that circumstance and can give protection against that kind of, for that group of people. So it's a very exciting area. Of course, as I've indicated, lots and lots of work has to be done. And if I could show just the next slide. One of the issues that we have is we're going to have to be very careful about our antibody engineering. And in Singapore, A*STAR and Chugai Pharma Body have been collaborating for a number of years on the production of neutralizing antibodies for the dengue virus. And we've learned a huge amount. We've been supported by GHIT, the Global Health Initiative, and this antibody has progressed a long way. But in that process, we realized we have to design the antibody so that it doesn't cause this thing that we called antibody-dependent enhancement. So far, I've told you only good things about antibodies, but this is a potential bad thing about antibodies. So some antibodies that are made against dengue virus, actually, when they bind the virus, they don't neutralize it. And because there's a receptor for antibody on the surface of some target cells, they actually help the virus to infect those cells. So this is very bad news. And as we go forward with both vaccine design and with antibody therapeutic development, we're going to have to be aware of this issue and make sure that our therapeutic antibodies don't do this, and also that the vaccines that we design don't induce this kind of antibody response. So this is a very important issue, but one that I think, fortunately, we're very aware of, and the work that was done in A*STAR and Chugai Pharma Body on the dengue project has given us a real insight here into how to engineer the Fc region of the antibody so that neutralization will happen, but this kind of antibody-dependent enhancement won't. Next slide. So I want to just briefly talk about immunity certificates. So this is a very important area, and the concept is superficially very attractive. So say you had a good antibody test, then you could tell, in theory, who couldn't get infected anymore. And those people could go back into society, help everybody do their jobs, could be safer working around patients, for example, in the hospitals. And so the idea is attractive, but as we'll see, there are a lot of caveats, and the WHO and Johns Hopkins and others have been talking about this because it has been raised as an issue amongst a number of governments, and I think it needs to be thought through very carefully. So at the moment, I think the honest truth is we absolutely don't know enough. Remember, this virus only appeared in December. So we don't really know what happens in the long run. So are people who've recovered fully immune, and how long will they be immune for? And is that immunity caused by antibodies, or is it, for example, held in the T cell compartment of the immune response? So we don't know these basic things. So we can't really assume that because you've got an antibody positive test that you really are protected and safe to others. And one of the worries that we have is that people might assume that they're safe to a second infection, and they might not take public health advice. So at the moment, our philosophy that's working is that everybody should assume they're infected, so they don't spread things to everything else. So everything we're doing, our behavior, is all to assume that we're infected. So the circuit breaker breaks the circuit of infection. And so the worry is that if you had such certificates, they might actually increase the risk of continued transmission. And I want to contrast that with a vaccination certificate. A vaccination certificate is altogether a good thing, because if you think about yellow fever, for example, a very good vaccine, vaccination certificate, you know, the person has volunteered, they have been vaccinated, the certificate says they've been vaccinated. So that's good. It's very different from an immunity certificate. And if we go to the next slide, I think it's also the last slide. I mean, these are the other things that are, of course, critical here. So obviously, patient confidentiality and privacy, access to regular testing, to know that if this is immunity, that it's still there. There's all issues about equitable access, who already we can see in some countries that even access to the PCR test is very non-equitable. You can imagine all sorts of bad things happening. So, you know, people are people. So some sort of black market for immunity cards might arise, and you would get people having risk of blocking employment and rejecting those who are not immune. And also what happened in the past, you know, people went to chickenpox and yellow fever parties to get themselves infected so they could get back into the workforce. So none of those things do we want. So I think, though the idea is an interesting one and has future potential, we'll have to go forward extremely, extremely carefully and with a great deal of thought about how we were to implement any kind of idea of this sort. I think that's probably the last slide.
That is the last slide.
Great.
Thank you, David, for that very valuable lesson. I feel like I've learned a lot. This is actually the fun part now. We're going to go into, we're going to give you a little bit of a break, actually.
Thank you. I'll have my tea.
Absolutely. We're going to launch a poll because I'm just curious who is listening in today. So I encourage everyone to submit their responses. What is your industry background? Are you from biotech? Are you a student, an academic? Are you a journalist? Are you from the government, or are you not willing to let us know? Others. Let's end this in 3, 2, 1. Okay, and I'm going to share the results. It seems like 38% of our viewers today are students and postdocs, followed by biotech and pharma industry professionals, academics, and healthcare professionals. Okay, that's interesting. And now I want to go to a second poll. This is a sentiment analysis poll. Just for fun, tell me, how confident are you that pharma and biotech companies will be able to develop an effective treatment or vaccine for COVID-19 in the next 12 months? I'm curious if our viewers today are the pragmatic sort or the optimistic sort. Let's find out. Okay, and it's almost, we have more than 800 people voting right now. In 3, 2, 1. Okay. I think we have a bell curve, don't we? Most people are on the fence, you know, the safest answer, and 52% are somewhat confident, and the rest are either very confident, good for you, and not confident at all, boo-hoo. So that was fun. Let me move back to our screen. All right. So that was fun. also in the registration stage, David, we had hundreds of questions that were submitted by the audience, and I combed through them, and I found a couple of them that I thought were interesting and that were repeated multiple times. So one of them was, Sir David, what is your opinion on convalescent plasma therapy? Philippine hospitals are starting to use convalescent plasma therapy for the critically ill.
Yes, so I think it's a very good question. I mean, clearly what we're expecting to have happen here is exactly what we're trying to do with the monoclonal antibody. That is to use the neutralizing antibody present in a patient to help another patient. I think, I mean, this has risks associated with it. Clearly, the donor person has to, has to have a willingness to do it, and then there are issues about this. I mean, we used to give many years ago horse serum for tetanus treatment. A horse was immunized with tetanus, and this is called a passive therapy, and it worked. But the problem is that you can make antibodies against component in the tetanus theory. Now, in this plasma donation, it's human to human, so there's less problem, but I suspect there will still be potential problems of this type, where you might make some antibodies to the incoming plasma, and this can lead to a condition called serum sickness. However, having said that, these are desperate times, and I think it's clear from anecdotal work that this has worked, and there's no practical reason why it wouldn't work. I think if it seems to be very effective, it will give a lot of confidence to the antibody teams as they develop their monoclonals, which will substitute in the end for this kind of process. But yes, so I think it's not something you would normally advocate, but these are not normal times, and I think it's something that is definitely worth considering. Like everything, we can probably get to do it better. So how we prepare the plasma, how we purify it, what we do, all of those things can probably be improved. But I think it's worthwhile pursuing for sure.
Okay, thank you. Another one that was interesting, actually quite controversial as well. Sir David, what are your views on herd immunity?
Yes. So, I mean, I saw a very nice article about a woman who had her children had immune deficiency disease, and she said, I'd prefer to call it community immunity, because she made the argument the fact that everybody else is being vaccinated against measles is what's keeping her children safe. So I think there's no doubt that this is important, but what we realize is that you have to have a very large number of people in the population be immune for this to work. It doesn't protect the individual, it protects the population from transmission. So in herd immunity, a child is still going to get measles. It's just not going to spread that measles infection to other children as easily, because all the children around them are going to be non-receptive to the virus host, essentially. They're not going to replicate the virus. So it's an important part of what happens when a vaccination program is very successful. I think, however, people have been very optimistic about this in certain countries. I won't say where. And we're not seeing that. One idea was that there were a lot of people infected and they were getting better, and therefore a lot of the population was essentially immune. And that is apparently pretty clearly not the case. So, there were one or two very early studies in a few places where a very bad antibody test was used, and they got false positive results. Just to say a few words about that, I mean, some of the early tests that came out, they were not sufficiently specific, and a lot of people have antibodies to COVID viruses because it's related to the common cold.
Yes.
And I know from my own experience as well that ELISA tests can be pretty bad. I mean, they work very well in the lab, but when you're dealing with something as complicated as human serum, people are hyperimmune to many, many different diseases. People have very variant levels of immunoglobulin in their blood. So you can get a positive test in those kinds of tests much too easily. So I think this is one of the problems. So we have to be very careful about this. But in the long run, of course, it's a great thing. But it does depend on everybody who's fit and able getting vaccinated, so that we can protect the people in society who are not able to be vaccinated. So yes, I mean, I think we're all passionate about vaccination now, which is good. We should have all been passionate about vaccination before, but we should be now. And herd immunity is one of the benefits of excellent vaccination. But You know, if we're going to have to make 4 or 5 million doses of vaccine, billion doses, you know, we've got a long way to go before this becomes an issue.
And another question that I thought was interesting was, are there any differences in the epitopes of current COVID-19? I guess they should have said SARS-CoV-2 strains. How would these then affect vaccine and therapeutic drug development?
Yes, so this is a very important question. We know with the seasonal flu, it's a kind of game. The flu virus is evolving and we have to get a new vaccine strain every year or two to keep pace with the virus. So everybody who's, Sebastian and others, and many people looking at these virus genomes are studying this question very carefully. It's a little bit early to say. The virus certainly isn't mutating at a huge rate, but there are mutations. And of course, at the moment, we don't have immunological pressure on the virus because it's just like, wow, I can infect everybody. so it doesn't have a problem. But as you know, people become immune, then variants of the virus that escape the immunity will be selected for. So we have to be very aware of this. And we don't know whether, you know, COVID-19 will end up being like some viruses, like smallpox, where we've been able to eliminate it because the vaccine is so good, or whether it will be more like flu, where the vaccine is incredibly useful, but it's not enough to completely suppress the virus. So those kinds of experiments are, you know, and results will come out from the studies that are being done now, but it's a very, very good question and a very important area. So far, I would say sort of so good, but it's really very early to say.
Oh, okay. This is something that you probably will be very interested in. How fast will we be able to scale up bioproduction to meet antibody titer requirements for, you know, clinical use of these antibody drugs?
This is a huge question because you know, we need to make a lot, and production of antibodies has improved enormously in recent years, and you know, we get cultures now that make 7 grams of antibody per liter of culture, which is amazing. But it's still a big job. I mean, purifying antibodies is not easy. It's a complex process. Obviously, you have to maintain, you know, complete sterility. The antibody has to be soluble and stable. We have to do a lot of tests of safety. So it's a big, big job. And I think it's much harder than making a vaccine. So that's definitely an issue. But it's not insuperable. And a lot of pharmaceutical companies have very big plants making antibodies now because, as I said earlier, you know, Humira, Herceptin, they're made in huge amounts. So there's a possibility. And in fact, quite a lot of manufacture happens in Singapore, in fact. So there's large manufacturing sites for antibodies here in this country. Interestingly, a very potent neutralizing antibody, the amount that we would need to use from our calculations with dengue is not great. I mean, if you think about using an antibody for like Herceptin, you know, we have to give it to the patient repeatedly. But with this kind of antibody, we're expecting we probably only have to give it once, and then it would give a period of protection. So though the manufacturing will be a challenge, we're not going to have to make so much per person as we would for the others, but we have to make an awful lot for an awful lot of people. So yeah, yes.
Well, it's 5:45. David, we've since run out of time. Thank you so much, David, for joining me as a guest on our Expert Opinion webinar. For those of you who enjoyed our session today, we will be publishing this webinar on our Asian Scientist YouTube channel later this week. So please subscribe to us to stay connected. On a personal note, David, it has been great to see you, virtually of course. I really look forward to seeing you and Birgit after the circuit breaker ends in June, hopefully. Thank you, David. Thank you to all of you who joined us this evening. Signing out from Singapore. Goodbye.
Bye. Thank you, Julia.
Bye. Bye.